Hello my dear students and welcome back to Excellence Batch, and I am your Diksha ma'am. So we have started here the chapter Biotechnology and its application. In the previous class we have discussed about the applications of biotechnology in various fields, and we have started with the agricultural part. In agriculture I have taught you that how the biotechnology is very important, and we have also discussed some few examples that how the biotechnology is being a boom in increasing the nutritional quality of the plants like golden rice. And we have also discussed how it's helpful in pharmaceuticals, where we have produced hirudin from a mustard plant, and also we have discussed about a pest resistant plant known as BT cotton, that how BT cotton itself can be resistant to the pest. Fine, you don't have to spray the pesticides on that. So today we are going to continue with agriculture, and also we'll be doing medicine. But let's talk about another pest resistant plant. So the another pest resistant plant is your tobacco plant. So as I've told you, why are we using biotechnology? Because there was a problem, and biotechnology is a kind of a solution. So what was the problem? So problem is tobacco root knot disease. So there is a disease which occurs to the root of a tobacco plant, and this is how the tobacco plant looks like when it is diseased. You can see this one is a typical plant. It's a typical type of a control plant where some organism has infected the tobacco plant and it is diseased. Fine. And this is a plant transgenic which is getting better, or which is pest resistant. Okay, so this is what happened to the tobacco root. Now who does this? Who is the causative agent? A nematode. The causative agent for this disease is a nematode. And obviously there will be a name of a nematode. The name is Melidogyne incognitia. Melidogyne incognitia. So that's a nematode that basically infect your tobacco plant's root and cause a root knot disease. Alright, so if this was a problem, why not to make this plant resistant to this nematode, which is a pest? So for that we used a technology which is RNAi. RNAi. I stands for interference. This is also known as gene silencing. So like I've told you, whenever we have to modify a plant, either we can add a gene or we can subtract a gene. So addition of a gene, you all know how we do it. We take a gene, we put it in the plant with the help of a vector. Right, but how can we subtract a gene, or how can we silence a gene? Basically we are not just removing the gene, we are silencing a gene. It's not like you take a piece of gene and put it out. It's not like that. You are silencing the gene. Okay, this is what gene subtraction is. So that's why this technology is also known as gene subtraction. So this technology is naturally present in our cells. Yes. So this technology was discovered, and when it was discovered we got to know that this is a kind of a defense mechanism of all eukaryotic cells. So naturally in your cell, in my cell, all this technology is present, right? So it acts as a natural defense. It acts as a natural defense present in eukaryotes. Now, why do we need this defense? We are not getting infected by the Melidogyne. Then what are we infected by? We may get infected by some viruses, so that viruses may not hijack our machinery all the time. So we have this technology. So this is for eukaryotes against viruses. Now how this technology works, first of all we'll talk about that what is present in our cells, that is a gift to us. All right, so for us, or in our body, the DNA is always double stranded, but RNA is single stranded. I think you all know that in our body RNA is single stranded and DNA is double stranded. So if somehow by mistake our RNA is double stranded, imagine this is RNA. If somehow our RNA gets double stranded, then what will happen? Now, ma'am, who is producing double-stranded RNA? No one is producing double-stranded RNA. Then how can double-stranded RNA be formed in our body? See, imagine this strand of RNA is a natural RNA produced by your body. Produced by your body or our cell, right? Imagine the second strand comes accidentally either from viruses or transposomes. What are transposomes? These are jumping genes. Your maximum DNA or your genes, they are like your junk DNA. They are not producing anything in your body. Rather, whenever they have to multiply, they multiply via some RNA intermediate, and when they multiply via RNA intermediate, they can jump from one place to another. This you will study in the botany also. Okay, so these transposomes, sometimes when they are multiplying, they produce RNA, right? So when they multiply via RNA intermediate. So this RNA sometimes accidentally, if it is complementary to your natural RNA produced for certain purposes that need to be produced, then at that time they can get complementary, they can join and they can form double-stranded RNA. Or somehow, if our one strand of RNA matches with some virus's RNA, then also it can get double-stranded. So this is a kind of a bad thing for you that should not be produced, because this is somehow is blocking your natural RNA that need to produce certain protein or have to do certain function. So at this time a machinery gets started in our body. A machinery starts, and under this machinery an enzyme is activated known as dicer. Dicer comes from word dicing. So the purpose of this dicer is to chop this double-stranded RNA into small pieces, right? And this is known as small interference RNA, siRNA. So basically this enzyme will convert your large double-stranded RNA into small interfering RNAs. Si means small interfering. Once they are formed, another set of enzyme will get activated, and the name of that is RISC. Imagine this is the enzyme RISC. RISC is the one which will help you in cancelling out this double-stranded RNA or destroying this double-stranded RNA, right? So the name is RNA-induced silencing complex. RNA-induced silencing complex. So this is another enzyme. So what this enzyme does is it will pick any of this small interfering RNA. Imagine it has picked this small interfering RNA, and it will destroy one of its strand and keep one strand for the reference. It will, RISC is the enzyme, it's enzyme complex that can only activate with the small interfering RNA. It cannot directly activate with the large double-stranded RNA. Right. So dicer is one which is very important here. Only if the dicer is present, only then the RISC can get activated, because RISC get activated with the small interference RNA, not the large double-stranded RNA. Okay. So now it will keep one strand of the small interference RNA, other it will destroy. Now it will roam around inside your cell and it will identify this strand of double-stranded RNA. Imagine one of the RISC is present here like that. Now it has 5' to 3' strand. Okay. And we have this RNA that's roaming around. Okay, double-stranded RNA. And it has identified this strand because it is complementary to this one. And now it will destroy, the RISC will destroy double-stranded RNA. So this is how you can destroy this double-stranded RNA, which is not supposed to be formed in our body, with the help of certain enzyme like dicer and RISC. Fine. So somehow we got to know that you can silence any of your gene in your body. Now ma'am, we have not talked about gene, we are talking about mRNA. So when you have studied in molecular basis of inheritance, you must have studied that what is a gene expression, what the gene is producing, and what gene is producing mRNA. So if any gene has to perform its function, it will go through the stages. DNA will form RNA, RNA will form protein, and protein is the one that do something. Okay, so if we block the stage mRNA, the gene's function technically is blocked, right? So by this technique, we are also blocking the RNA. Why not to use it for our purpose? All right, so how did we use it? So we took a tobacco plant. All right, imagine this is your tobacco plant. All right, so in tobacco plant, we added two genes. So genes were added. What are these? Genes added in plant. Okay, so now we have added two genes. Say it be gene 1, say it be gene 2. Because this is genetically modified organism, so genes will be added. Okay, so here we have added gene 1 and gene 2. Gene is what? The segment of DNA. Now these segment of DNA we have produced in such a way that one gene will produce sense RNA, other will produce antisense RNA in the tobacco plant. Okay, and we have done this very cleverly. We have added gene in such a way that one will produce sense strand, other will produce antisense strand, and both these strands are complementary to each other. If they are complementary to each other, you all know that they will join. Fine. How does melidogyny infect your plant? Whenever melidogyny have to infect your plant, imagine this is melidogyny. Okay, this is a nematode that is infected. Whenever it has to infect your plant, this will produce mRNA. Okay, so what we did, we added genes in a way that one gene will produce mRNA exactly equivalent to the gene of melidogyny. So imagine this sense RNA which is produced is equivalent to the mRNA of melidogyny. Now these two are both complementary to each other and they will form double-stranded RNA. Once the double-stranded RNA is formed, then RNA interference will be started and a RISC will be activated which have the strand complementary to the strand of your melidogyny's mRNA. As a result, it will find the mRNA of melidogyny and destroy it wherever it is present. Imagine we have produced this plant. In this plant, imagine this antisense RNA is RNA that is equivalent to the mRNA of nematode. Okay. All right. So now these two strands, because they are complementary, because they are complementary, they will form double-stranded RNA, and double-stranded RNA formation is very important if you want to start RNA interference. Once the RNA interference is started, RISC will be activated, and RISC has a habit to take one strand and destroy the other. And now what RISC will be doing? For example, this nematode is infecting you. So this nematode, when it will infect you, will send this strand 5' to 3' strand, right? So now what our RISC will do? Okay, just a minute. Now what our RISC will do? This RISC which have the complementary strand of this one, imagine 5' to 3', will identify it and destroy it. So basically the all purpose of this technique is to activate a RISC in a tobacco's plant which have complementary strand of the mRNA of nematode, so that if nematode comes and infect you, the RISC is already have been activated to destroy it. Just like when you take vaccination, right? In vaccination you produce antibodies in your body, so that if the organism is coming inside your body, you can destroy it because you already have activated the antibodies. In the vaccination, what you do, you take dead killed organism so that the immune system get activated. In this one, we are taking the mRNA of the organism so that a double-stranded RNA and entire this thing will be activated, which is RISC. So RISC is just like antibody of our body, right? All right. So this is how we can make our transgenic plant. Transgenic, what are transgenic? Any plant which have their genes modified, you call them as transgenic. Okay, now let's read the NCERT of this, because this is important, otherwise you will never be able to understand. Several nematode parasites parasitize a wide variety of plant and animal, including human being. So just up to this stage, you all knew that nematodes infect the animals. But yes, nematodes are also infecting the plants. Okay. So a nematode, Melidogyne incognitia, infect the root of tobacco plant and causes a great reduction in yield. So at which part of the plant it is attacking the root? As a result, the yield reduces. A novel strategy was adopted to prevent this infestation, which was based on the process of RNAi. RNAi takes place in all eukaryotic organisms as a method of cellular defense. This method involves silencing of a specific mRNA due to a complementary double-stranded RNA molecule that binds to and prevents translation of mRNA. So what we are doing? We are silencing a specific mRNA, like mRNA of nematode. Okay, how are we doing it? By producing a complementary double-stranded RNA. Fine, and as a result, the translation of the mRNA will be stopped. How does Melidogyne infect you? From DNA it produces mRNA, mRNA produces protein. Protein is the one that destroys the plant. So that formation of protein from the mRNA is known as translation. So technically, if the mRNA is blocked, translation will not take place. The source of this complementary RNA could be from infection by viruses having RNA genome or mobile genetic elements, transposomes that replicate via RNA intermediate. So in our body, what is the source of the another RNA? Because we never produce two RNA, we never produce double-stranded RNA. The second RNA can be from a virus or from these jumping genes. Jumping genes are also known as mobile genetic elements. Okay. Now let's see how we incorporate that into the plant. Using agrobacterium vector, nematode-specific genes were introduced into the host plant. As I've told you, we have added the genes in the plant. How have we added? By using agrobacterium. And as I've told you, these are nematode-specific genes. That means one of the gene will be producing the mRNA that will be equivalent to the mRNA of nematode. This is what means nematode-specific genes. Okay, so all right. The introduction of DNA is such that it produces both sense and antisense RNA. So it has produced both strands of RNA in the host. These two RNA being complementary, now they will become double-stranded RNA and then it will initiate the RNAi. Once RNAi is initiated, RISC is activated and then that RISC will silence the specific mRNA of what? Nematode. The consequence was that the parasite could not survive in a transgenic host expressing specific interfering RNA. So as a result, the nematode will not be able to survive in the plant. The transgenic plant therefore got itself protected from the parasite. Because it is getting protected from the parasite or a pest, it is pest-resistant plant. Okay, that's how we have used a very complex technology for our one solution and make it simple. In RNAi, the genes are silenced using double-stranded DNA, double-stranded RNA, single-stranded DNA, single-stranded RNA. This is the most common question. Simple. Always double-stranded RNA. Okay. Next. Tobacco plant resistant to a nematode have been developed by introduction of DNA that produced both sense and antisense RNA, particular hormone, and antefeedant, a toxic protein. Simple. Our transgenic plant produces both sense and antisense strand. We have added a nematode specific gene which will produce both the strand. Both being complementary will join and produce double-stranded RNA. Which part of the tobacco plant is infected by Melidogyne incognita? Very simple, the root. Answer is 4. Next, RNA interference is used for which of the following purposes in the field of biotechnology? Okay, why are we using it? Why are we using it? To develop a pest-resistant plant against infestation by the plant, that's the first answer. Because we have to make plant pest-resistant. Not for mineral uses, not to reduce post-harvest loss, and to produce a plant, not to produce a plant abiotic distress. So answer is 1. Okay. Alright, next. So the next is, before studying the medicine, let me give you one more example in which we have used the RNA interference, and that is flavor saver tomato. Flavor saver tomato. This is one kind of a variety of tomato which are transgenic. That means their genes have been modified. Okay, so what's the problem in tomato? The problem in tomato, okay, let me drink some water, then I'll tell you what's the problem. The problem in tomato is that whenever you go to market to buy a tomato, you always want that your tomato should be firm, red, juicy. But the problem is that when the tomatoes are transported from farm to the market, it takes some time, and in that time interval, these tomatoes, they ripe, like they overripe, and they are not at its good quality, and nobody purchase it, and it get the entire yield, whatever has been produced, the food has been produced, the tomato that has been produced, it got wasted. Fine. So the problem was that our tomato should remain firm for a longer period of time, that they should have their more shelf life. What is the shelf life? That you can keep it on the shelf for a longer duration of time. That means you can keep it and they will not get overripe. So that's a problem. So first we find out what makes the tomato overripe in a short interval of time. So they got to know that there is one enzyme. There is one enzyme, and the name of enzyme is polygalacturonase. So this is the enzyme. This enzyme destroys pectin in cell wall of plant. Right. So if the cell wall is destroyed, the tomato gets soft and overripe. Right. So the problem was this enzyme. That this enzyme, the one which is making it overripe in a shorter duration of time, because it is destroying the pectin in the cell wall of the plant, and the tomato become very soft. Okay. So if you know, this is the enzyme. And you all know enzymes are usually protein, and this enzyme is produced from mRNA, and mRNA is produced from a gene. If mRNA is involved, why not to silence it? Okay, so the problem was over ripening of tomato. Over ripening of tomato. They have low shelf life. So how did we increase the shelf life? We produced new tomato known as flavor saver, in which we silenced mRNA that produced this enzyme polygalacturonase, which destroyed the pectin. Once this is silenced, pectin will not be destroyed and your tomato will be firm and nice. Okay, so this was the thing that they did. So if it is silenced, if mRNA, then there will be no polygalacturonase, no pectin destroyed, and tomato will be firm. Okay, so actually the problem was they also these farmers, they also came up with the another solution. So if they knew that the tomato get overripe in short duration of time, they started plucking the tomato when they are not ready to be plucked. Right, that had also a disadvantage. What was that? That it will be little green, they will be not good, they will not be sweet, right? They will be not be good in taste and texture. So the problem was, you can't pluck it before time, you can't pluck it even when it's time, because when it's time, after that they will get overripe. So they came up with these solutions, not the farmers, the scientists, obviously, right? So that's the flavor saver tomatoes. Now let's talk about application of biotechnology in medicine. So in medicine, the first application of biotechnology is production of recombinant therapeutics. Production of recombinant therapeutics. What are recombinant therapeutics? First of all, what are therapeutics? Therapeutics are all those substances that are produced in your body. but somehow they are not produced, then less in number, that causes you disease. Let me give you an example. For example, if a person is not producing growth hormone, and that growth hormone is a therapeutic, and when if you are producing it with the help of biotechnology, recombinant DNA technology, that is known as a recombinant therapeutic. So biotechnology is again a boom here. Why? Because these all these therapeutics, you cannot take it from another person. For example, if one person is not producing insulin, you can't ask other to donate your insulin. It's very tough. Fine. So by biotechnology, you can produce as much as you want. There is easy production and there is mass production of these therapeutic, which can protect people from a number of, or they can cure person or people from number of diseases. Fine. Till now, we have produced 30 recombinant therapeutics all over the world. 30 recombinant therapeutics have been formed, out of which only 12 are marketed in India. In India, we only use the 12 out of 30. Right? Let me give you an example of very important therapeutic, that is genetically engineered insulin, and sometimes it is also known as humulin. Now, why is used to produce this insulin? All right. So there is a disorder known as diabetes mellitus. In diabetes mellitus, the person have low insulin or no insulin. Right. In any ways, the person's glucose metabolism is disturbed, because the function of the insulin is to regulate your glucose metabolism to reduce the blood sugar level. If you do not have the insulin in your body, then person will have high glucose in blood. And that is a disease known as diabetes mellitus. In general way, people also use the term known as sugar, that a person is suffering from sugar, they have sugar, right? So diabetes mellitus is a disorder. Diabetes mellitus is a disorder. In this disorder, the person is not producing insulin at all or in a very low quantity. As a result, the glucose metabolism is not maintained and the blood has high level of glucose. Okay, so when the recombinant insulin was not invented, what people used to do at that time? The people used to have option, that was they took insulin from pancreas of slaughtered animals, of slaughtered pig and cattle. This was the option they had, the only option. But this has a side effect, they causes allergies. So they were also not good. If a person is having a therapeutic to get rid of the this disorder, maybe that can be fine, the disorder can be relaxed, right? It can be can go away. But the another thing that get heightened was allergies, because this is a foreign protein, this is coming from a pig or a cattle. So there was a need to produce the exact same insulin, which is a human insulin. Fine. So the interesting point about the human insulin is that our human insulin is obviously, it's a protein, it's a hormone made up of protein, is produced from gene. Gene produces mRNA. mRNA produces protein, right? And this protein is insulin. But insulin, when it is formed in the form of proinsulin. What is a proinsulin? Inactive insulin, which is formed by, or which is formed of combination of three type of chains of amino acid. All right, let me show you a diagram if it is there. Yes, in the NCERT you must have seen this diagram. The insulin, when it is formed from RNA through translation, it is formed in this manner. It is formed in this manner. This is A chain, this is C chain, and this is B chain. Right? Okay, let's move back to the simplest diagram. A chain, C chain, and B chain. Later on, this proinsulin is converted into a free form of insulin by post-translational changes. So post-translational changes is the character of eukaryotic cell, not a prokaryotic cell. So after changes, what used to happen? The A chain gets separated, B chain again gets separated. Both are joined by disulfide bonds. Which bonds? Disulfide bonds. Okay, this is A chain, this is B chain. A chain is of 21 amino acid and B chain is of 30 amino acid. And the C chain is taken out, which is of no use. C peptide chain is of no use, it is taken out. And this is the pure form of insulin. Okay. Right now in biotechnology, whatever we were doing with the gene cloning. In gene cloning, we used to use vector and that goes inside your bacteria, that is E. coli. And bacteria, they are prokaryotes. So if I try to produce my insulin in E. coli, these post-translational changes cannot occur because they are the only character of eukaryotic cell, not prokaryotic cell. So there was a problem. You understand the problem? If I try to produce the insulin by putting the gene in E. coli, then I cannot produce it because that E. coli will not be able to perform this, whatever it is done. Right? So Eli Lilly, an American company, they came up with the idea. What was the idea? They used two E. colis. They performed two types of gene cloning, or two setups were done. Okay, experimental setup. So this was E. coli 1 and this was E. coli 2. They said if E. coli cannot perform this, why not to give only few part of a gene. One gene that was added in E. coli was only producing A peptide chain. Another gene that was added in E. coli 2 was only producing chain B. So what they did, they separately produced two different genes. So this was the gene that was producing A peptide and this was the gene that was producing B peptide. So this is the gene for A peptide and this is the gene for B peptide. Now this E. coli will be producing your A peptide chain and this one will be producing your B peptide chain. Now they join them by forming disulfide bonds in them, because disulfide bonds are the bonds that are present between the two chains of insulin. And this is how the Eli Lilly, an American company, produced insulin separately in two types of E. coli. Fine. So let's read NCERT, the things will become more clearer. The C peptide is not present in the mature insulin and is removed during maturation into the insulin. So this is your mature insulin. Fine. So this insulin is also known as mature and proinsulin is immature insulin. Okay. The main challenge for production of insulin using rDNA technology was getting insulin assembled in a mature form. So main problem was that the prokaryote cannot do post-translational changes. So how we do it? So Eli Lilly, an American company, in the year 1983, what they did, they prepared two DNA sequences corresponding to A and B chain of human insulin. So they produced two genes. Okay. Introduce them into plasmids of E. coli to produce insulin chain. Introduced into the different plasmid. Chain A and B were produced separately. They were produced separately. They extract them and combine them by the disulfide bond to form human insulin. And human insulin, thus it was invented. And there was a time when it was invented, a lot of people were dying from diabetes mellitus. So when they came to know, oh, they have produced, they went to the hospital, they give injection to all the patients, and patients, they were not dying, rather the process was getting reversed, right? They started living, they were alive. Fine. So this was how, this was kind of again a nice gift by the biotechnology to the human beings. Next, let's do question. Which of the following statement is not correct? The proinsulin has an extra chain peptide called C peptide. True. Proinsulin is immature insulin which have C chain. The functional insulin has A and B chain linked together. True. Genetically engineered insulin is produced in E. coli. In man, insulin is synthesized as a proinsulin. Okay, I think that question is little incomplete. So here there is a sentence linked together by hydrogen bonds. So that's how the statement is incorrect. I'm so sorry, it was a typological error. So yes, it is produced in E. coli, and yes, in human it is produced as proinsulin. Okay, all right. Next is the two polypeptides of human insulin are linked together by a very simple question. Which bond? Disulfide bridges or bonds? Answer is 4. Next, all right. So gene therapy we'll be doing in the next class. We'll be doing that in the next class. All right. So that's it from my side from the medicine and agriculture. The next part of the medicine we'll be discussing in next class, and that's the gene therapy. I've already shown you, and your suspense has been broken. So stay tuned and keep on watching the entire video so to complete this chapter, and I'll meet you in the next class. Bye bye.